in your syllabus, theory syllabus, right? Various chromatography techniques. So very fastly we'll see its introduction, followed by its general principle and technique, and finally its application. So as you know, this is one of the important technique for the identification and separation of mixtures of chemical compound into its individual components. Further, it is a form of liquid chromatography, and it consists basically two phases, right? One is the mobile liquid phase, and second one is the solid stationary phase. So based on different rate of interaction between the solutes and the stationary and the mobile phase, they will promote its separation, right? So this involves the distribution of components of a mixture to be separated between two phases. So the component of the mixtures are partitioned, okay, as this is a type of liquid chromatography. Okay, so mixtures will be partitioned between adsorbent, that is your stationary phase, and your liquid, that is your solvent mobile phase. Now all the compounds have different solubility, okay, and different adsorption to the phases between which they are to be partitioned. So individual substance is based on their relative affinities towards stationary phase and mobile phases, and this will be the responsible factor for the TLC separation. So what kind of stationary phases can be used in TLC? So as adsorbent, usually silica gel or alumina that is coated on a plate, and mobile phase, that is a developing liquid which flows through the stationary phase carrying samples with it. Now components having more affinity towards stationary phase, so of course they will travel slowly, right? And vice versa, if you see the components which have less affinity towards the stationary phase, so they will move very fast on stationary phase. So here if we discuss the method, so as an adsorbent in stationary phase, inorganic materials like silica gel, kyselgur, aluminum silicate, bentonite, this can be used, and as an organic material, cellulose, acetylate, charcoal, activated charcoal, dextrine gel, and polyamides can be used. Solvents, that depends on the, of course, that depends on the solubility of your analyte or solute. So petroleum ether, benzene, carbon tetrachloride, chloroform, methanol, ethanol, ethyl ether, variety of solvent you can use, and that you can decide that you can decide your particular compound will be soluble in which particular solvent, right? And selection of this adsorbents and solvents, having this three, adsorbent should not, I think three criteria, like adsorbent should not adhere to glass plate, right? Solvents should be of high purity. Now selected based on the, this selection is based on the nature of the compounds to be separated, whether it is polar or non-polar, and based on that you have to select your mobile system. Now what we generally do, we evaluate retardation or retention factor. This indicates the position of migrated spores on the chromatogram. So in TLC, the results are represented by RF value, which represents the migration of solute relative to the solvent front. So the equation, as you know, distance traveled by the solute, this is divided by the distance traveled by the solvent front. So if we develop a TLC technique, so step number one for the development of this TLC technique is the preparation of slurry, right? That is your stationary phase. So how you will make this slurry? So a plastic or a glass or aluminum sheet, right? So on which you will do coating of with a thin layer of silica gel, that is your adsorbent. So plates must be, and this plates must be dried, activated, and stored in a desiccator until used. After that, preparation of saturation chamber, you can call as the preparation of tank, where you will fill your mobile phase. So solvent mixtures should be freshly prepared for analysis. Now solvent is poured downside of the tank, that is approximately to the 1.5 centimeter depth, and tank is covered with the glass lid and kept for saturation, right? And in third step, application of sample means spotting of your sample. So a very small amount of sample solution to be analyzed is applied in a small spot with a capillary tube, that is 1 centimeter from the bottom of the TLC plate. Now this is how it actually this instrumentation, we can say instrumentation or setup for experimental setup for TLC. So you can see this is your saturation chamber, or you can say mobile phase well. Here in a tilt position you have position your TLC plate, and this is your TLC jar, this is your jar cover, and this is wet filter paper, just to show whether it is your mobile phase chamber is saturated or not. And this is your TLC plate and how your compound is traveled on this plate. This is simple setup for TLC experimental setup. Now this TLC is developed in a chamber which contains the mobile phase, as you can see. When the mobile phase rises up the plate up by capillary action, okay, that is opposite to the gravitational force, the components dissolve in the solvent and move. So, individual compounds in the sample move up at different rates. Now, as you know, the light dissolve like based on the light dissolve like principle, more polar analytes interact more strongly with the stationary phase, and it will move very slowly, while non-polar analytes interact less strongly with the polar silica gel, and more strongly with the less polar mobile phase, and moves higher up. So once the solvent reaches the top, that is below 1 to from top side, that should be below 1 to 2 centimeter of the TLC plate, the plate is removed from the developing chamber and position of the solvent front is marked, just like here you can see, right? Now this is the separation of different compounds. Here you can see mixture of three components, here two and here again three. This is the same here. This is actually three, but spotting is not properly. There is some concentration overloading, or sometimes this is volume overloading. You can say if you do proper spotting, then see. And here, if you can see, there are more proper way spotting is done, right? So the solvent is allowed to evaporate from the TLC plate, and as the compound is colorless, it can be visualized by a suitable method, like for lipids, iodine vapors you can use, or for drugs which are colorless, for that you can use iodine vapor. Then amino acids, for amino acids, neon hydrant reagent that can readily form the complex with the amino acid and gives a color complex. And further, if your compound is colorless and if you want to still visualize your analytes, separated analytes, then as I discussed in theory classes, manganese activated zinc silicate, right? So that is a fluorescent compound which is added to the adsorbent along with the silica gel. So because of this addition of this compound, this will allows the visualization of the spots under a black light, at which UV wavelength is 254 nanometer, right? So once visible, the RF value of each spot can be determined. This is a simple way how the different compounds has migrated at different distance, and this is the solvent front, that is 5 centimeter. So you can see this is compound A, B, C, D, U1 and U2, means unknown 1 and unknown 2, and this is the standards. So you can see by dividing simply with the solvent front, you can evaluate your RF values, right? So results of TLC and RF value, qualitative results of TLC expressed as fractions of 1, right? So it can be expressed from RF values like RF into 100, no more than 2 decimal places. Now, RF values can be used to aid in the identification of a substance by comparison to the standards. So, how qualitative measurement can be done? If you have some standards of that same sample, so by comparing its RF value, you can identify what is your unknown compound. Now, comparisons should be made only between spots on the same sheet and run at the same time. Further, identical substance will have the same RF value, whereas the non-identical compounds will differ in their RF values. Now, what are the applications? So, this can be used for qualitative and quantitative analysis to separate organic compounds and to test the purity of compounds. Further, it is useful for the separation of lipids, amino acids, sugars, etc. And further, it is useful for the identification of components of mixture. Then it can, following the course of reaction, analyzing the fractions collected during purification, analyzing the purity of the compound. These all tasks can simply be done with TLC experiment. So this all things you have already studied in your theory classes. Now, if we see the practical section, so here you can see, is my screen visible?
Yes, ma'am. Yes.
Okay, so this is what TLC separation of dyes. So here in this practical, two dyes, that is rhodamine B and fluorescein, the mixture of this two dyes was separated on the TLC plate. So for this solvent system, that is a mobile phase, we have used, that is the mixture of acetone and water in 3:3 volume. Okay, and solution that we have prepared for dyes 1%, right? But 1 gram in 100 ml of solution, that is what, 1% solution, right? Then we have prepared this mixture, and this is the resolution factor, and here this is the calculation for rhodamine B and fluorescein. Okay, so I have one video so that you will have some clear idea about dyes, how you can separate on TLC plate. Just a minute. Mm, okay, just assume here, instead of paper, the procedure will be remain same for thin layer chromatography, where alumina set which is coated with the silica gel. Okay. Just a minute. Okay. I'm dyes that is mostly separated on paper chromatography. So instead of dyes, you can see the separation of amino acids and drugs. Okay. So again, I'm sharing my screen here, and yes.
Separation of amino acids by TLC. Amino acids are the building blocks of peptides and proteins. They possess two functional groups, the carboxylic acid group that gives the acidic character and the amino group that provides the basic character. The properties provided by
Is this video audible?
Yes, ma'am.
Yes, ma'am.
these groups are used to characterize the amino acids. Here, TLC or thin layer chromatography can be utilized to identify the different amino acids. Separation occurs as each component, being different in chemical and physical composition, interacts with the stationary and mobile phases to a different degree, creating the individual spots on the plate. Here, our aim is to identify an unknown amino acid by spotting it along with the known amino acids. Apparatus: TLC plate, solvent, a mixture of N-butanol, acetic acid, and distilled water in the ratio 12:3:5, 3 known amino acids, an unknown mixture of amino acids, ninhydrin, a spray bottle, a blow dryer. Developing a TLC plate requires a developing chamber or vessel called TLC chamber. This can be as simple as a white mouth jar. The chamber should contain enough solvent to just cover the bottom. It should also contain a piece of filter paper or other absorbent material to saturate the atmosphere with solvent papers. Finally, it should have a lid or other covering to minimize evaporation. Procedure: cut the plate to the correct size and using a pencil, gently draw a straight line across the plate approximately 1 centimeter from the bottom. Using a capillary tube, a minute drop of amino acid is spotted on the line. Allow the spot to dry. Spot the second amino acid on the plate. Repeat the steps to spot the unknown. Place the plate into the TLC chamber as evenly as possible and lean it against the side. Allow capillary action to draw the solvent up the plate until it is approximately 1 centimeter from the end. Remove the plate and immediately draw a line across the solvent front. Dry the plate with the aid of a blow dryer. Spray the dry plate with ninhydrin reagent. Dry the plates in hot air oven at 105 degrees Celsius for 5 minutes. Here, ninhydrin will react with the spotted amino acids and the resulting complex, which is colored, makes them visible on the plate. After some time, mark the center of the spots, then measure the distance of the center of the spots from the origin and calculate the RF values. Some tips for a successful result. Make certain that the spots apply to the plate or above the surface of the eluting solvent. Hold the plates by their sides. The solvent front must not advance up to or beyond the edges of the plates.
So this is how actually the amino acids that you can separate on TLC plate. Now I'll show you the video for
हेलो एवरीवन, आज के सेशन में
separation of drugs.
This video will cover experiment number 12, the TLC analysis of analgesic drugs. It will go through the procedure as well as data and observations. A reference plate and sample plate were each made and analyzed. The procedure is shown on figures 1 through 7 and 10 for the reference plate and figure 8 for the sample plate. Many of the procedural steps were the same, and so pictures were not repeated. Data and observations for the reference plate are shown in figures 9 and 11 and 12 and 13 for the sample plate. The solutions used on the reference plate and sample plate are shown here and throughout other slides. To start, a TLC plate was used and a line was drawn with a pencil 1 centimeter from the bottom of the plate. On the 1 centimeter line, 6 small dash marks were made and numbered 1 through 6. Above are the 6 solutions that are used on the reference plate. Notice that a wooden pencil is used for this, not a mechanical pencil or a pen. This video is showing the spotting technique used with the reference plate. A capillary tube was used. It is a small tube that is open on both ends. It is dipped into the solution and then lightly touched onto the TLC plate where the dash mark meets that 1 centimeter line. The solvent is allowed to evaporate, and then the solution is spotted one more time to make sure that enough of the active ingredient is present on the TLC plate. Figure 4 shows the reference TLC plate after it has been spotted. Figure 5 shows the developing chamber and developing solutions that were used. The developing chamber was just a large beaker, and the developing solution is 0.5% glacial acetic acid in ethyl acetate. The developing solution was poured into the developing chamber, so only about half a centimeter deep was used. Figure 6 shows the reference TLC plate in the developing chamber. A recrystallizing dish was used as a lid. There's an arrow pointing out the current solvent level. It is about 1/3 of the way up the plate. The TLC plate was removed when the solvent was about 1 centimeter from the top. Figure 7 shows the plate after it was removed. After it was removed, a pencil was used to draw the line where the solvent reached, and then the solvent was allowed to evaporate. The same procedure was used for the sample plate. A TLC plate was obtained, a line was drawn with a pencil 1 centimeter from the bottom. These 4 analgesic drug solutions were spotted on the dash marks on that 1 centimeter line. The plate was developed in 0.5% glacial acetic acid and ethyl acetate and was removed from the developing solution when the solvent was approximately 1 centimeter from the top. In order to view the data, 2 methods were used. First, the plates were observed under UV light. This is the reference plate under UV light. Second, the TLC plates were placed in a jar containing iodine crystals under.
This is how iodine chamber looks. This is a jar in which iodine crystals are there. If you put your TLC plate inside of this jar, so there will be iodine vapor inside of this jar. So because of this, your colorless compound will be looks like this yellow spots, right?
Very low heat. You can see the jar in figure 10. Figure 11 shows the reference TLC plate after it was removed from the iodine jar. Figure 12 shows the sample TLC plate viewed under the UV light, and figure 13 shows the sample TLC plate after it was exposed to iodine in the iodine jar.
Okay. So I hope you are clear with this TLC, TLC of amino acids, TLC of drugs, analgesics drugs have been separated on TLC plates, and of course dye, that is a mixture of rhodamine and fluorescein. So on YouTube there are some videos that is available for separation of dyes, but that is on paper chromatography, not on not on TLC plate. So exactly as you have seen in the separation of drugs and amino acids, we'll do the same. We'll do this experiment in the same manner, but instead of dyes and drugs, we will spot dye. Instead of amino acids and drugs, we will do the spotting of this dyes. But for visualization of dye spotting, there is no any method is required, because dye itself having some color, so you can easily identify at which particular distance the that particular dye has migrated the difference on TLC plate. Okay, so am I clear now with this TLC experiment? Yes. Do you have any doubt now? Okay, I'll share the both the PPTs and the YouTube links on MS Team, so you can see the link of the different practicals, and you can search by yourself if you find some other good videos for the same practicals. Okay. So now this practical session is open for questions. If you have any question, you are free to ask.
मैम नीन हाइड्रेन रिएजेंट क्यों यूज करते हैं?
Yes, very good question. See, नीन हाइड्रीन जो अभी थोड़ी देर पहले आपने वीडियो में देखा होगा, वो क्या होता है, मेनली दिस इज अ वन काइंड ऑफ केमिकल दैट फॉर्म कॉम्प्लेक्स, कलर्ड कॉम्प्लेक्स, राइट. सो वो बायोलॉजिकल जो भी मॉलिक्यूल्स है, राइट, प्रोटीन्स है, एमिनो एसिड्स है, मोमेंट इट विल सी एमिनो एसिड इटसेल्फ कलरलेस है, तो उसके साथ वो क्या करेगा, एक कॉम्प्लेक्स फॉर्म करेगा, जो अभी थोड़ी देर पहले अपनी वीडियो में देखा था. एंड द मोमेंट वो कॉम्प्लेक्स फॉर्म होगा, दैट विल बी कलर्ड. दैट्स व्हाई नीन हाइड्रीन यूज एज अ विजुअलाइजेशन मेथड, one kind of visualization method. Okay.
ओके मैम.
Anything else? पूछिए कुछ भी. दैट आपने वो जो देखा नीन हाइड्रीन में ना, दैट इज पर्पल कलर, राइट. सो दैट इज वन स्पेसिफिक रोहमान पर्पल कलर जिसको बोलते हैं, है ना. बिकॉज नीन हाइड्रीन मेनली जो एमिनो एसिड्स के अल्फा एमिनो ग्रुप्स होते हैं, प्राइमरी एमिनो एसिड्स, है ना, उसके साथ रिएक्ट करके एक पर्पल, जिसको एक स्पेसिफिक बोला जाता है, रोहमान पर्पल, राइट. So the chromophore formed is the same for all primary amino acids, and the intensity of the color formed that depends on the number and chemical nature of the amino groups being analyzed. कितना तुम डार्क हो रहा है, तो वो उस पर डिपेंड कर रहा है कि कितना इंटरेक्शन है, ऐसा, राइट. यस, यहाँ पे कोई चैट में है, डाइस में उसी कलर का स्पॉट मिलता है. यस, आई मीन जो स्पेसिफिक डाइस का होता है वैसा. यस, यस, मेहुल, आपका क्वेश्चन है कि डाइज में कैसे रहता है. हाँ, यस ऑफ कोर्स. जैसे कि एक है डाइज रोडामाइन बी जिसका पिंक कलर रहता है, एंड एक है फ्लोरेसेंट तो जिसका फ्लोरेसेंट येलो होता है ना, वैसा होता है. तो वो क्या होता है, तुम जैसे ही बीच में उसको स्पोर्ट करोगे, राइट, मिक्सचर का उसको स्पोर्टिंग डाला, तो आप देखोगे कि रेड कलर वाला नीचे रहेगा और येलो कलर वाला ऊपर रहेगा. उसको कोई विजुअलाइजेशन की जरूरत नहीं है क्योंकि उसके पास खुद का एक ओन कलर है, वो खुद क्रोमोफोर कंटेन करता है. दैट्स व्हाई उसको कोई विजुअलाइजेशन नहीं चाहिए. अगर आपको सबके सामने पूछने में ऐसे है तो यू कैन यूज योर चैट बॉक्स. ओके. एनी आयोडीन चैम्बर आपने देख लिया, नीन हाइड्रीन आपने देख लिया, नीन हाइड्रीन सॉल्यूशन. और कोई, और भी सारे बहुत सारे विजुअलाइजेशन मेथड्स है, ऐसा तो कुछ नहीं है. दिस इज लाइक इनऑर्गेनिक प्रैक्टिकल्स आप करते हैं ना, ये डालने से ये वाला कलर होता है, ये पीपीटी जनरेट होता है. सो स्पेसिफिक कंपाउंड कोई स्पेसिफिक कंपाउंड के साथ इंटरेक्ट होके दे विल प्रोड्यूस अ कलर. ओके, सो दैट कैन बी यूज्ड एज विजुअलाइजेशन मेथड. एनीथिंग एल्स? नहीं है तो मैं कुछ क्वेश्चन पूछती हूँ. यस, वन मोर क्वेश्चन इन चैट बॉक्स. जस्ट अ मिनट. क्या लिखा है? मैम, कैन वी यूज बोथ साइड ऑफ टीएलसी प्लेट फॉर सेपरेटिंग टू डिफरेंट मिक्सचर्स साइमल्टेनियसली? सो नो, ऐसा नहीं होता है. बोथ साइड मतलब फ्रंट एंड बैक? नो, दैट्स नॉट पॉसिबल. एक साइड पे तो आपका एक जो एल्यूमिनियम फॉइल, जो एल्यूमिनियम का सीट रहता है, उसके ऊपर आप कोट कर सकते हो. दोनों साइड से कोट करके तुम लोग क्या करते हो, टिल्ट करते हो, है ना. टिल्ट पोजीशन में यू आर पुटिंग योर टीएलसी प्लेट इन द जार. अब क्या होता है, दिस सिलिका इज लाइक वेरी फ्रेजाइल, है ना. तो क्या होता है जार में, वो समटाइम्स कैसा होता है कि समटाइम्स सिलिका कैसा होता है, इट विल कम आउट फ्रॉम योर, वो जो सपोर्ट आपने यूज किया होता है. अगर सिलिका जो अपने जेल, उसके ऊपर पूरा स्लरी, जो अपने एक इवन स्लरी आपने एक लेयर बनाया है, if somehow it will come out from your TLC plate, so what will happen, this will completely affect your separation. तो बोथ दी साइड, ऐसा नहीं होता है कि फ्रंट और बैक, नो, ओनली वन साइड पे दिस इज पॉसिबल. और इन केस चलो कर भी लिया साइमल्टेनियसली, बट यू विल हैव टू टेक मोर केयर, okay, in such a way that your silica adsorbent should never ever disturbed. Okay. Good. Any other question? अच्छा लग रहा है आप लोग इस तरीके से क्वेश्चन पूछ रहे हो. आई अप्रिशिएट फॉर दिस. देखो अब इसमें से कितने सारे क्वेश्चंस पूछ सकते हैं कि एमसीक्यू अगर आपका टेस्ट होता है प्रैक्टिकल का, तो ऐसा क्वेश्चन हो जाएगा कि लाइक if your compound is polar and your stationary phase is non-polar, okay, and your mobile is moderately polar, so what will be the rate of your how at what particular distance, sorry, which compound will be come out first from TLC plate or elute first from the TLC plate. इस तरीके से पोलैरिटी नॉन पोलैरिटी बेस में थोड़ा सा उसमें इधर उधर करके यू मे आस्क क्वेश्चन. देन एक क्वेश्चन ये हो सकता है, व्हाई in UV light black spot is observed on TLC plate, because in TLC you are adding what, fluorescent compound, fluorescent compound, that's why. ठीक है. फिर एक और क्वेश्चन ये हो सकता है कि by which action your solute will migrate on TLC plate to the upside. तो कौन सी एक्शन आएगी, कैपिलरी एक्शन, ग्रेविटेशनल एक्शन,
Capillary action.
capillary action. Okay. And then which kind of adsorbents you can use. तो वो जो सारे नाम थे ना, केशलगर थे, जेनोलाइट था, वो सारे नाम थे, सिलिका था, राइट. तो वो सारे नेम आपको याद रखने हैं. एक और क्वेश्चन ये हो सकता है कि Yes, why ninhydrin gives a color with the amino acid complex? Because alpha amino groups के साथ क्या करता है? It will form a complex and that gives a room of purple color, right? And और क्या question हो सकता है? Yes, what is the equation for retardation factor? Okay. और भी आप सोचो उतने बन सकते हैं, तो है नहीं, बोलिए और कुछ. You can use your chat box. Please feel free to ask question. बाकी तो बहुत सारे. This is very simple practical TLC. जो पेपर क्रोमेटोग्राफी में करते थे that exactly you will do on what TLC प्लेट पे करोगे बस. This is the difference. यहाँ पे वहाँ पे cellulose था as a stationary phase. This will act stationary phase, and here silica gel will be the used as stationary phase or it's adsorbent, right? पूछिए.
मैम,
Yes.
हम हमको कैसे पता चलेगा कि हमें कौन सा स्टैंडर्ड लेना है?
हाँ, like कौन सा? अच्छा, in case of your unknown compound.
यस मैम.
See, in see, when you are analyzing your unknown compound, then there will be no any standard. You have to do characterization of that unknown compound. But if suppose you have compound, like in that particular compound, if suppose there are two or three things, okay, but you are sure there is, suppose in your mixture there is a caffeine, okay, and other than caffeine or two three compounds are present, right? So if you want to particularly identify caffeine in your mixtures, so you can use caffeine as a standard, okay. And yes, and as you ask the question for unknown compound, so suppose for unknown compound you are getting your RF value, suppose 0.67, okay. So TLC would be the preliminary technique which will give you a rough idea about your qualitative analysis. If it is 0.66, 0.67, and at 0.67 if you are matching in the library, so there is one compound at 0.67. Then further you need to confirm by using the another technique. If at 0.67, suppose there is one particular tryptophan, one kind of amino acid at 0.67 retardation factor, right? So library has give initial answer. Then after performing TLC, you have to do UV analysis. Then you have to perform NMR. Then if you have to perform IR. And for this all studies, if you are get the similar results as you are getting with the library, then you can confirm this unknown compound is exactly the same as we have initially matched in the library. Okay. ओके, आपको समझ में आया मैंने क्या बताया आपको?
हाँ मैम.
हाँ, यस. एक और क्वेश्चन है कि इन सम केस स्पॉट इज इन रेक्टेंगल, so what is the concept behind it? See, ideally, yes, rectangle in mainly in HPTLC, so you will get. See, there is a difference between the TLC and HPTLC. HP जहाँ TLC में राउंड स्पॉट्स होते हैं, वहाँ इन HPTLC में रेक्टेंगल होता है, लाइंस होता है, एक लाइक हॉरिजॉन्टल लाइन होता है, और उसमें उसी तरीके से वो डेवलप किया जाता है. Or else, if you are asking about rectangle spots in TLC, so that TLC will be erroneous. But if you're talking about rectangle spot in HPTLC, so that is the correct way of spotting of in TLC in HPTLC, right? वो आपने अपने डिफरेंस देखा होगा कि हाउ द शेप ऑफ स्पॉट इन TLC एंड HPTLC. ओके, आप चाहो तो HPTLC का वीडियो यू कैन सी ऑन YouTube कि हाउ वो इस तरीके से कैपिलरी से वो रेक्टेंगुलर शेप में उसका स्पॉटिंग होता है. बट इन TLC वी जनरली डू राउंड स्पॉट्स बिकॉज हम मैन्युअली एक कैपिलरी लेके सिंपली करते हैं. बट दिस इज द कंप्लीट ऑटोमेटेड वर्जन, इंस्ट्रूमेंटल वर्जन ऑफ TLC. Okay, any other question? Just a minute. Okay, you can say this one if you have some time. Yes, here you can see how this coating is
Carbonite Gero, a name that is synonymous with the design and manufacture
rectangular
of high-quality furnaces and ovens.
High performance or pressurized thin layer chromatography. It consists of mainly three parts: sample injector, visualizer, and scanner. Then nitrogen gas chamber. It is connected to the sample injector. This pointer should show the pressure in between 4 and 6 for effective sample loading.
Yes, here you can see how this coating is done. It begins to load on
It automatically allows the syringe to load the sample. It is the chromatographic
the
plate.
way rectangular spotting is done.
Plate size varies with the sample numbers. These plates are pre-coated with silicone over the aluminum sheets. It may need activation if it is stored for a long time. After it reaches moving to park position, remove the injection. Essential oil components will evaporate. Keep it in visualizer at 254 nanometer. It is at 366 nanometer. If the sample is not of non-UV observing type,
See how
it should
the
be
spotting
immersed in
looks like
H2SO4 for
rectangular.
derivatization, but it should not exceed 10 seconds. And again air dry. The bands are appear in it.
Okay, anything else? Okay, if you still have any question, you can